So this discussion involves the intersection of molecular biology, which it's basically biochemistry, and genetics, a pivotal area for treatment refinement. I'm going to clarify some basic issues and define the area that I'm going to be discussing. But first, I'm going to tell you that this is the work of Dr. Amy Yasko. I work with her. I do the clinical management of the people who come to us. She does the constant research of the literature, handles test processing, and makes recommendations for supplementation. This is the link to a presentation she gave to another doctor's group, and it contains a lot of background information about methylation and methyl groups and her source materials and her references. So you get two for the price of one when you tonight and when you work with us. I'm going to give you some background, and then I'm going to launch into clinical application. Dr. Amy has a tremendous amount of material online, so you can ease in to what she does. It takes a while to get enough background to understand how to use her method. It involves dragging out your biochemistry books and relearning pathways you probably have forgotten. You really can't do it if you have a busy practice and you like to sleep at night. I managed to do it because I had been in psychiatry and I had time. But yeah, you also have to like biochemistry. So I put these links into the presentation so you'll have them when you get the PowerPoints. Okay, on the home page of my website, there's an opt-in for an ebook which can give you in-depth information about methylation and methyl groups as simply put as I could make it. Then you can layer other information in on top of that. Chapter 3 spells out the process of making methyl groups, the biochemical pathway that I'm going to be talking about tonight. And this is important for integrating how to use this material clinically. Okay, this is by way of review. I'm going to go over these issues, these topics, so we can be sure that I can be sure that you understand what I'm talking about. DNA is composed of a strand of nucleotides. The nucleotides are 4. Adenine, the A, let's see if I have a cursor. Yes, I do. Adenine, the A, cytosine, the C, guanine, the G, and thymine, the T. Genetic information exists in the sequence of these nucleotides. Genes exist as stretches of nucleotide sequence along the DNA chain. And what this means is this. This is the DNA chain, and say if you started here and ended here, the sequence of the bases, the CGAT, the sequence of these bases is genetic information. And if one of those bases is out of sequence, say there's a G when there should be a T right here, that will have an impact on the function of this strand of DNA, whatever it's supposed to make. So the gene part is, say, from here to here, and there is a C here instead of a T. That will impact whatever the product of this DNA is supposed to make. Okay, let's see. I'm going to just go on. Okay, here are the 4 bases. These thymine, adenine, cytosine, and guanine. Now, by themselves, if it's just this molecule, that's just called a base. But thymine and a pentose sugar, a 5-carbon sugar, that is a nucleoside. with an S. Oh, where's that? Okay, adenine with a pentose sugar, cytosine with a pentose sugar. Those are all nucleosides. If they have a phosphate ester attached to the pentose sugar, that links them together. The chemical bond is in the phosphate ester, and that actually comprises the rail. Like if a strand of DNA is like a twisted ladder, the outside of the ladder, not the rungs, but the outside, is actually called a rail. I asked the fireman. Anyway, so this sugar and the phosphate ester is the rail, and these guys are the rungs. Okay, so let's see. This is a picture. This is a depiction of the pentose sugar. This is ribose. It has an oxygen and a hydrogen molecule on the 2-carbon. And here is deoxyribose, just a hydrogen on the 2-carbon. And that's how DNA and RNA are named. I'm really not going to talk about RNA at all tonight. I wanted to streamline this. So that's it. This is deoxyribose, and that's the sugar that's in DNA. And then there's ribose, and that's the sugar that's in RNA. Okay. A single nucleotide, I actually talked about this already. A single nucleotide polymorphism, or SNP, is a change in one base in the gene. A SNP may, in fact, impact the function of the product of the gene, depending upon its sequence, where it is in the DNA base sequence. It also, SNPs may not make any difference at all. When you get 90,000 SNPs from 23andMe, some of those SNPs have not been characterized, and they may not do anything. Okay, genes encode for enzymes. An enzyme may function more slowly as a consequence of a SNP, which downregulates the enzyme activity. Or it may function more rapidly, which is an upregulation. SNPs are considered mutations in the gene. SNPs can be important, especially when they impact the function of genes that produce critical enzymes in important biochemical pathways. Mercifully, the gene and the enzyme it produces have the same name. The name of the enzyme always ends in ASE. Dr. Amy's genetic profile focuses on SNPs that impact the function of key genes in a very important intersection of biochemical pathways in which methyl groups are made. A methyl group is a 1-carbon, 3-hydrogen molecule with critical functions. They are small but mighty, like water, or oxygen, or nitric oxide. Think about it, these tiny molecules. Methyl groups are like the traffic lights of your biochemical pathway. They move some reactions along and stop others. They change molecules from one substrate to another. They are pivotal to the function of many body systems. In the list of online support links I showed you earlier, there are videos of lectures that go into detail on each of these functions. It's not possible to do it tonight. Plus, I wanted to be sure to cover the additional features of Yaska's work that make it more effective clinically, notably how she uses lab test findings. Methyl groups have a global function in biological organisms that is not necessarily conveyed by this list, except by the items repairing and rebuilding DNA and RNA, inflammation, and energy production. These give you a sense of the global and overarching aspect of methylation. DNA methylation is a crucial modification of the genome that is involved with regulating cellular processes. These include cellular processes involved with embryonic development, transcription of DNA, chromatin structure, X chromosome inactivation, and chromosomal stability. Consistent with these important roles, a growing number of human diseases have been found to involve aberrant DNA methylation, and these are some of them. So I am going to continue on. We have, 60 minutes, and but Dr. Amy does go into some of these. Okay, so Jasko is interested in regulatory molecules, molecules that regulate the function of other important structures. Methyl groups regulate the function of genes. They silence a gene or prevent it from expressing. This is a depiction of a methyl group attaching to or methylating a gene. This is epigenetic regulation, regulation that comes from a molecule in the gene's environment. Epi means around. It is not something within the gene itself that impacts the gene's expression. Epigenetic regulation is inheritable. In 2003, experiments were done at Duke University which demonstrated the potency of DNA methylation for altering the physical characteristics of an organism. Agouti mice have a gene that gives them yellow coats and a propensity for obesity and diabetes when it is expressed. That's this guy on the left. One group of pregnant agouti mice got a diet rich in choline, betaine, folic acid, and B12, substances that support methyl group formation and or are outright methyl donors. Another group of genetically identical pregnant agouti mice got standard mouse rations. The B vitamins and methyl donors given to the fortified mice caused methyl groups to attach more frequently to the agouti gene, thereby silencing its expression. So, without changing their DNA structure, the fortified agouti mothers were able to produce healthy brown pups, this guy is the normal one, that had a normal weight and were not prone to diabetes. This happened strictly by an epigenetic process, methylation. Okay, so now we're into the biochemical pathways that produce methyl groups and some of the aspects that certain SNPs can have on this pathway, on these pathways. This is a sample test report from Dr. Amy's genetic test. Dr. Data offers this test for her, and Genetic Profiling System runs it, I believe. Doesn't matter. This is a blood test. The blood can be obtained by a finger stick. There's another test, a survey genetics test that we often use, which is the saliva test from 23andme.com. Blood is more accurate than saliva, but it is considerably more expensive. What I have seen from saliva testing is that it's not so much that the SNPs are profiled inaccurately, as it is that important SNPs are either not found, you get a N/A, no answer result, or they're not on the test in the first place. So blood is better, but saliva is a considerable savings. If you have a really complex chronic illness and you can afford the blood test, that's a better way to go. If you have a really chronic complex illness, you may want to do both. But okay, so here we are, and what I'm looking at is the intersection of 5 chemical pathways involving methyl group formation, neurotransmitter production, and the excretion of toxic metals. This is a stripped down version of this pathway. Let me see if I can, yeah, I did. I got rid of it. It wouldn't move before. Okay. Anyway, this is a stripped down version of this pathway without all of the vitamins, cofactors, and minerals that are necessary for its function. I'm going to start here in cycle 4. Cycle 4, this guy, is the methionine cycle. Methionine gets adenosylated and becomes S-adenosylmethionine. S-adenosylmethionine gives away its methyl group for tremendously important functions like DNA and RNA formation, protein, methyl groups are involved in both the formation and the deactivation of neurotransmitters. Lipid metabolism, especially the lipids in your brain, tremendously important functions that you can hear about. I just want to get into the clinical business here. So SAMe gives away its methyl groups and becomes S-adenosyl homocysteine. S-adenosyl homocysteine then loses an adenosyl molecule and gets down here and becomes homocysteine. Now, homocysteine has 3 possibilities. Where did that? It can go become methionine again through this BHMT pathway. Boy, that cursor is just leaving me after seconds. Or it can go through, this is what Dr. Amy calls the shortcut route, and this is the first thing we activate. We try to get methyl group production happening right away. And then this is what we call the long route, and we work on getting that to happen. And this is the transsulfuration pathway. Okay, so this BHMT pathway, if there, we look, Dr. Amy's test looks at 4, I'm going to just keep moving this cursor because otherwise I lose it. She looks at 4 genes in this pathway. 3 of them are a downregulation, BHMT 1, 2, and 4, and BHMT 8 generates an upregulation. This acronym is the name of a gene that produces an enzyme that is upregulated in the case of one SNP and downregulated in the case of the other 3, BHMT 1, 2, and 4, and 8 is an upregulation. Okay, so what we try to do is we use phosphatidylserine complex, not Serophos. Serophos has a whole gram of material in there that is a proprietary blend. We don't know what the heck is in it, and we're very careful in this protocol. We're treating really touchy people, kids who have not been able to recover from autism and adults who are chronically ill, and really touchy people. So we are paying attention to the first and the second molecule, like, for example, magnesium citrate. We would want to know that the person doesn't have too much citrate on a metabolic analysis profile or an organic acid test before we would give mag citrate. We would give something else. We might give ionic magnesium. We're extremely careful about what we use. So anyway, Dr. Amy uses phosphatidylserine complex, which contains phosphatidylserine, phosphatidylcholine, and phosphatidylethanolamine. And it has 1 or 2 other things in there, but that's okay. Anyway, so we are trying to get homocysteine to get a methyl group from trimethylglycine, which is, hang on. Trimethylglycine, which is betaine, gives its methyl group up and becomes dimethylglycine, and then homocysteine is back up here being methionine again, and then it goes around, it does this cycle again. Okay, So the other option here, let me see, the long route, that is the route that goes through MTR. This is actually an acronym for methionine synthase, and MTRR, methionine synthase reductase. Now, the long route works together with this cycle, cycle number 3, which is the folate cycle. Okay, so when the methionine cycle is working in the clockwise direction, the folate cycle is working in the counterclockwise direction. So tetrahydrofolate is going down here and becoming 5,10-methylene tetrahydrofolate, which if I'm going to just cut, I need to get this done in 60 minutes. So all I'm going to tell you about this SHMT gene is that if there's a mutation in this gene, it is going to grab 5,10-methylene tetrahydrofolate and send it out and make it into a purine, which is necessary. I mean, we need them for DNA and RNA production. But for our purpose, we would like to keep it in this cycle and send it down here so that MTHFR can work on it and produce and turn 5,10-methylene tetrahydrofolate into 5-methyl tetrahydrofolate. So, and then 5-methyl tetrahydrofolate goes up here in a manner of speaking, and attaches to methionine synthase. I'm going to give you the short version. Methionine synthase holds onto it while MTRR works to keep the B12 in the area in its reduced state, and basically they exchange the 5-methyl tetrahydrofolate, they put it back on the homocysteine, and methionine is made again, and then it goes through the cycle again. Now, the issue is that there are many more genetic interferences with using the long route than there is with using the shortcut route. So we get the shortcut route going first while we're working on getting the long route activated. If you have a mutation in this gene, it causes you to dump minerals, especially lithium. And lithium is really necessary for the uptake of B12 and folic acid into the cell, which is where this happens. It happens in liver and kidney and certain brain cells. A SNP in this gene is an upregulation. A SNP in this gene is a downregulation. The primary job of MTRR is to keep the B12 in the area in a reduced state, because then it can work. Okay, so the big gene that everybody's talking about is over here. It's MTHFR, methylene tetrahydrofolate reductase. There is a mutation at the C677T point that is a problem. You can't turn 5,10-methylene tetrahydrofolate into 5-methyl very well. And then a plus homozygous mutation here is really a problem. That person is going to have something, they're going to have issues, health issues. But the wonderful thing about all this is that you can just supplement 5-methyl tetrahydrofolate. However, the rest, in order to have your body function well, you can get by this block, but usually by the time you've spent half a lifetime with a block here, you have things off in your gut, and you have other factors that make it but and the mutations around this gene, this SHMT mutation and the MTR and the MTRR, may give you other health issues that make it difficult for you to feel well. All right, so that's the story with MTHFR C677T. Back over here, homocysteine. Homocysteine, as I said earlier, we wanted to be able to activate that shortcut route using homocysteine. But if there is, we wanted to be able to activate the shortcut route, and we wanted to be able to send homocysteine up through this long route for making methyl groups. But if you have a CBS mutation, this is an upregulation of the genetic activity here. So when this is upregulated, and for example, this may be downregulated, or there may be a block over here, then homocysteine will tend to pool. And then especially if this guy is upregulated, it will grab homocysteine, send it down this transsulfuration pathway, and what will happen, it will unbalance this pathway, and then you will get the production of taurine instead of glutathione. And glutathione, as you're no doubt aware, is a very important detox and antioxidant molecule. Okay, so let's see. Let's see. I'm going to point out that this is a representation of neurotransmitter formation. Tryptophan turning into serotonin, tyrosine turning into dopamine, and then COMT, catechol O-methyltransferase, needs methyl groups to break down especially dopamine and norepinephrine. You know, I think I'm just going to move along at this point because I would rather have time left than no time at all. This is a urine amino acid test, and I'm going to just review for you what we think when we see this test. When we see a high ammonia level, if I go back, you can see ammonia is right here. And Dr. Amy used to think that high ammonia had to do with this CBS pathway being overactive, but at this point, she is thinking that it has to do with the flora, the organisms in the gastrointestinal tract. If we see high ammonia, we immediately think dysbiosis. Okay, then methionine. This is like the first among equals of amino acids. Having methionine below really is non-optimal. You would want, depending on the patient's state, you would want to supplement methionine a bit here. And I'll just say this, I'm not going to show you a slide of it, but if you get a urine amino acid test back and all of these amino acids are low, then you know that this person has trouble with their gastrointestinal mucosa. Either not eating enough protein or they've got gastrointestinal mucosa problems. Okay, so this methionine containing a methyl and a sulfur group and starting off that methylation process is really important. But what you see as you look further down here, this taurine is through the roof. So let me just go back. You see this. So you're coming down here. You just note homocysteine, cystathionine, cysteine, and taurine. We'll take a look at what the other values are. So this person is high taurine, and they're spilling their sulfur groups out of their body, and they're likely not making glutathione. And look at where cysteine is. Cysteine is super low. Cystathionine is on the next page, so just kind of remember it. Glutamine and glutamate look okay. Cysteine is low. Here's reduced cysteine, and they're both low. Anyway, and this is serine. It's part of the BHMT pathway that I just reviewed. And these guys are all low. This person needs phosphatidylserine, phosphatidylethanolamine, and phosphatidyl, there. This guy can use the PSPCPE that we work to start that BHMT pathway. All right, here's cysteine low, cystathionine low. Methylhistidine, this is high. You need methyl groups to break down histamine. So this person may be having trouble with allergies. So basically, once again, you're going to have to read and study if you're interested in this area. This is a paper that Dr. Amy and I wrote, and it's on my website, nancymullenmd.com, on the home page under publications. Okay, this is just to remind everybody, you are here. The little area that we're looking at, but Dr. Amy has a canny instinct for picking out important areas of biochemistry to focus on that give you a foothold for taking care of the rest of the problem in your body. Okay, so now I'm going to just take a look at how we regard lab testing, the way we use it. I'm going to talk about our using urine essential elements and hair elements analysis together in order to get a status, in order to get a take on the mineral balance in your body. It's also important because minerals function all along that methylation pathway. I didn't put them in because I wanted to simplify it, but vitamins and minerals are all along that pathway. Okay, so many of the slides in this section will have subheadings at the bottom right. Just ignore them. They are from another presentation. And when I show you a sample test result, just look at the substrate that I'm talking about. I generated these slides from sample test results, and I changed only what I needed to change to demonstrate what I was going to be talking about. What I'm going to be talking about is dumping, mineral dumping. You take a look at this urine test, and let's see. We use random catch urine tests. We don't use 24 hours. It's incredibly difficult. You can't get a 24-hour urine test from a child, and it's hard from an adult. But random catches work if you look at data from sequential tests. We collect these samples at around 5 or 6 p.m. You can see, okay, I'm looking at lithium here. You can see lithium looks fine. It even looks a little hot. But is it? Do we really know? Or is your body spilling lithium into your urine for some reason? For example, do you have an MTR mutation? It happens commonly in MTR mutations, but it can happen to anyone. important minerals can spill, such as potassium, zinc, molybdenum, selenium, and others. In order to answer whether lithium is in fact dumping or whatever, you compare the level of lithium in the urine essential elements to the level of lithium in the hair elements analysis. So here you are, lithium down here. Look at that, is not high at all. That lithium that you saw 1 minute ago in that slide is dumping, and this person is actually low lithium. So, all right. So this is what it looks like when your body stores of lithium are low, period. Here's lithium, low, and here's lithium, low again. That's it. Body stores are low. And this is actually, this method, Dr. Amy developed this method. It actually is more refined than blood testing. You can, of course, get a serum lithium level. If you do one in this patient, you'll find that it's low. The ranges for a serum lithium done at any lab are the correct ranges whether or not you are taking pharmaceutical lithium. Some clinicians have seen low lithium levels on their patients' serum lithium tests and have told their patients that serum lithium ranges are only accurate if you're on pharmaceutical lithium. This is not correct. Many chronically ill people are low in lithium. You can see this same phenomenon with any mineral. Given the importance of minerals for catalyzing chemical reactions in your body, it's good to know what this looks like in your patient's situation. Switching over to zinc, you'll see the same thing. Let's see, here's zinc. It looks high. When zinc is dumping, it will look high on a urine essential elements test. But if you look at hair, it's going to look low. You can get this same picture if you're supplementing the mineral. However, if you're supplementing zinc, you may see something that looks like dumping. Keep supplementing until the low value comes into the normal range, and then you can back off a little. Probably not remove the mineral supplementation entirely, because there's an indication that this person has a tendency to dump zinc. And this is what you'll see when your body stores of zinc are low, period. Low zinc in the urine. And, this should be a hair. Wait a minute. Okay, low zinc in the urine. And that, look, it should be a hair test, low in the hair. Dumping may have occurred in past, and now the body does not have enough stores to dump. You don't know what occurred unless you tested during the prior period. Okay, so now we're going to talk briefly about the gastrointestinal tract. This is good. I am right on target. The gastrointestinal tract could be an entire lecture in itself. Let's see, it's a the gastrointestinal tract is a central issue for the vast majority of chronically ill patients. It takes special care to treat, and the treatment has to go on over time. The function of the GI tract is to digest food and absorb the nutrients it contains. As such, the small bowel and large intestine have many projections called villi that act to increase the surface area inside your abdomen. These projections protrude into the lumen of the GI tract. Imagine this as the inside of a hose-like structure. It's a tube with protrusions inside of it that are brushing digestive enzymes onto the food as it passes. Flattened out, it has the surface area of a tennis court. So there are many spaces and folds inside your gastrointestinal tract within which good or problematic organisms can sequester. If you've got problem organisms there, you have to change your gastrointestinal environment and keep it changed over time to improve your situation. Microvilli are like a lawn. They get damaged easily if they don't have the right, you see it, damage, and this is the basis for leaky gut. They get damaged easily if they don't have the right conditions. In normal people, they repair easily also. In a lawn, you can have patches of slightly dry or yellow or outright dead grass or brown dirt. And the degree of repair differs for each of these situations. The same is true for microvilli. And the more chronic the problem, the more compromised the immune system, the more arduous the repair. The villi are damaged. Let's see. Okay, next. 70% of your immune system surrounds your GI tract and the gut-associated lymph tissue. Your GI tract is a very immune-active organ. Tomorrow night, you're having a lecture on ileocecal valve syndrome. This ileocecal valve area is really immune-active, and lymphoid nodular hyperplasia may be present there. These nodes, the lymph nodes from that area may become large and hypertrophied, like some of the giant pus-filled tonsils that you may have seen. But you just can't lop them out, as you may choose to do with tonsils. It takes a reduction in GI tract inflammation and careful maintenance of the GI environment over time to solve this. Obviously, patients with impaired immunity, secondary to methylation or genetic issues, will be at a disadvantage here. Immune function tops antibiotics. Improve immune function, and then antibiotics will work. If you do not win the battle in the gut, you're not going to win the war. Obviously, I am only scratching the surface of this topic here, but let me show you. Okay, now look, we get two different kinds of stool analysis. We get doctor's data, comprehensive stool analysis, and we get another test called the DRG, Doctor's Research Group, Pathogen Plus test to detect very specific, harder to find organisms like H. pylori. There are certain organisms that underlie dysbiosis, wrong organisms in your gastrointestinal tract. Like if you have a parasite, my experience is that about 50% or more chronically ill people have parasites, and that regular labs, LabCorp and standard medical labs will not find them to save their lives. Alternative labs, Doctor's Data and Genova and Metametrics, also pretty poor. We don't even use them to test it. She uses this DRG lab that uses DNA probes, and they only look for blastocystis, which is a problem. But I have another lab, a specialty lab, that is pretty good. It's not Parasitology Center. Parasitology Center is okay, but if you ask me, I'll tell you what this other lab. It's not a lab; it's a doctor, and his work is great. His parasitology testing is excellent. I really count on this person. However, his treatment, his name is Raphael D'Angelo. He's an MD in Colorado. His treatment is always essential oils, which is just, they're not that great. They don't work that well, and you have to get rid of those parasites. All right, so I'll tell you the other thing here. In this expected beneficial flora column, we do not want to see this much Bacteroides. Even though Bacteroides is normal flora, it just gets in there. We don't want to see Bacteroides. We use Usnea for it. We don't want to see E. coli. She uses ECX, which is one of her RNA formulas, which are good, but they're expensive, but that's because RNA is a new technology. So we use cranberry. And Enterococcus, we used to use sage. She's now using grapefruit seed extract. We make a formulation of herbs to deal with this. We don't really want to, this is a 4-year-old, so we don't really want to use antibiotics for him, although we could over here. But antibiotics, it's like too much. It knocks things off in the wrong direction. So for this 4-year-old, she would use these RNA formulations. I might use natromycin. You know what, though? I have learned not to second guess her. She looks at lab tests all day long, and I think she is taking into account that this little guy is only 4. All right, so she would use grapefruit seed extract and caprylic acid here for this. So what I would tell this mom to do is I would tell her to make a mixture of grapefruit seed extract, probably 1 cap, caprylic acid, and probably I would throw in one of Dr. Amy's natromycins, a cranberry, and Usnea is a liquid. We're going to have to use drops of Usnea. But I would make a dry mix of cranberry, grapefruit extract, and caprylic acid, and I would start him out with the tip of 1 prong of a fork of it once a day, alternating with probiotics, good high-dose probiotics. And I certainly would adjust this guy's diet. And this will go on over time with repeat testing. This little one also has H. pylori. So we're going to use Dr. Amy's peptimycin, tiny dose. That's why we would make a mix of caprylic acid, grapefruit extract, cranberry, and then Usnea, and probably a natromycin, and a peptimycin. Her mix for Helicobacter, and we'd start him out with just 1 fork prong tip of a mix of all those dry herbs, and then we build that up over time and see how he did. Sometimes it happens that in the middle of a treatment like that, he'll break out with a strep infection, and then I would give him the appropriate antibiotic, usually some kind of Biaxin or some kind of penicillin, and you hold the herbs in place and you give them at the same time that you're giving the antibiotic in order to hold down the growth of yeast and opportunistic organisms. But my main point here is, let's see if I can get to this. Okay, this next person is a 47-year-old man. And once again, look at this. If you just leave all this in, fine, just went after the Clostridia here. If you just leave all this in place, you're not going to get the gastrointestinal tract right. So I would use like a paleo diet or some appropriate kind of diet. And, she uses these RNAs. This is for Klebsiella. This is for strep, and this is for E. coli. And there's not much out there for E. coli except rifaximin, which, once again, okay, so she's recommending it knocks it. Rifaximin will do it, but you'll end up with a mess in another direction. It's just not it doesn't work that well. So I'd probably use a paleo, gluten-free, casein-free, paleo diet that also eliminates any food that this person is allergic to, and that is it.